is Dr. Michael Good from Griffith University in Australia, and he's going to talk about from using epitopes to hold parasites, other vaccine strategies to trick a parasite to give up.
There you go. Thanks very much. Okay. Thanks very much, Carol, and particular thanks to Dr. Agre and to Prakash and Audrey and Trish as well, of course, for organising the whole meeting, and delighted to be here, actually very much honoured to be here. And I'm going to talk about this topic here, which is an unusual topic, I suppose, how to trick a parasite, but I think that's what we have to do. The first half of the talk will focus on antibody approaches to vaccines, and the second half on cellular immunity approaches to vaccines. And the first half will be largely theoretical, but the second half will have, I'll be presenting a lot of data, some unpublished. So this is from the WHO site just recently. The number of vaccine antigens which have been currently active and those which are not active, inactive. In other words, they've been tested and are no longer being followed. So this is 40 years now after blood stage antigens were first cloned by David Kemp. And we're all disappointed that we haven't got a subunit vaccine by now. At the time, I remember Gus Nossel saying there'll be a vaccine in 5 years. That never eventuated, of course. But hearing some beautiful data from Simon and Prakash, we might see a subunit vaccine successful very soon. But that aside, I want to focus on some of the published data which shows limited protection with various subunit vaccines. And here we've got on the left, we've got a fragment of sera, and on the right a large fragment of MSP3. And as you can see, although there is some effect, this is in clinical trials, although there is some effect, clearly these people are not totally protected. These vaccinees are not totally protected at all. So what's the problem? What is the problem with subunit vaccines? Now, both Simon and Prakash have really stated the main issues, of course, polymorphism, quality of antibody, quantity of antibody. But I want to sort of just say that the real problem from where I sit is the design of the vaccine antigen. And I'll tell you what I mean by that and how we might be able to fix that, or at least address that issue. Currently, subunit vaccine antigens are designed to induce strain-specific immune responses, but malaria parasites are not homogeneous. So when I say they're designed that way, that's just a fact, that's an observation. They do induce strain-specific immune responses. So the question is, how do we target conserved epitopes of antibody-mediated immunity? These are themes that we've been hearing already. And I want to propose a slightly different strategy to what we've been hearing so far. So in thinking about this, I've sort of revised some of the characteristics of natural immunity. And we often say we don't want to copy natural immunity because it takes so long to develop. But maybe we do. We just want to find out what part of it we want to copy. So let's look at natural immunity. In children, clinical immunity is slow to develop in areas of high endemicity. In areas of high endemicity, adults have clinical and parasitological immunity, but this does wane if exposure ceases. Clinical immunity does not develop in areas of low endemicity. Agglutination of infected red cells by serum is a very strong indicator of, correlate I should say, of natural immunity. And clinical and anti-parasite immunity can be transferred to children very effectively using immune serum from immune adults. So the classical famous experiment of Cohen, Carrington and McGregor has already, in 1961 in Nature, has already been referred to. But I want to show this version here by Pierre Drouil's group 30 years later. And the reason I'm showing this is Pierre did it, they did it a little bit differently. They transferred the serum IV, so you got an immediate effect of the serum, an immediate effect which wasn't, it was delayed in the Cohen paper because they did it IM. The parasites seem to disappear, then they come back. But then they transfer back the same hyperimmune serum, or not hyperimmune serum, the same adult immune serum, and the parasite drops dramatically. So this is not due to the development of an escape variant. These are the same parasites. There's been no variation here. There just wasn't enough antibody to control these. So what is the antibody that's controlling them? This is a question. So this is one of the, to me, one of the most striking papers in malaria immunology by Marsh and Kevin Marsh and Russell Howard in 1986. And it shows to me something very, very interesting, and I love reading this paper. You're looking at the ability of convalescent serum from children who've recovered from malaria to agglutinate their own strain. This is from the Gambia. 10 children, as you can see, the convalescent serum agglutinate their own strain, but by and large, they don't agglutinate any other strain. So there's something in that convalescent serum which is relative to why these children have recovered from malaria, all coincident at the same time. But when you look at adult serum in the same village, now you see something very different. Now you see all the adult serum, essentially all the adult serum, can agglutinate all of the children's parasite isolates, the different isolates. So it's a very different picture. So the first take on reading that is that, well, probably this is what's happened. The adults have all developed antibodies to each individual variant in that community, in that village, and now they're protected because they've got antibodies to them all. But of course there is another possibility, and that's this, that maybe the adults have developed antibodies to a conserved epitope, which is present on all the parasite strains. We're talking about the red cell membrane antigens here, the variant antigens. So which is it? Well, if you go to table 3 in that paper, it's a very interesting table, I think. They took, now this is only from one adult, so one adult serum, so we have to sort of interpret the data cautiously, but certainly for this one adult, GA4, they absorbed a GA4 serum against isolate 340 and then eluted those antibodies off that parasite, off that infected red cell. And now they found that that antibody, eluted from 340, could agglutinate all the strains. So that is suggesting that in GA4 has developed an antibody to a conserved epitope present on all the strains. Now, that's interesting. I think that's very interesting. But equally interesting is the next row down. So when they absorbed GA4 with 340 but didn't elute the antibodies off, what they found was that GA4 absorbed with isolate 340 could not agglutinate any of the strains. So what that's saying is not only does this adult GA4 have these sorts of antibodies to a conserved epitope, but he or she doesn't have antibodies to any of the variant-specific epitopes at all. So let's go back now to a more recent paper published last year from Justin DiRichimo and with Patrick Duffy, where they looked at full-length VAR2-CSA, recombinant VAR2-CSA ectodomain, and they absorbed onto that multigravid serum, which is associated, of course, with protection against pregnancy-associated malaria. So this is a figure from the paper. So they absorbed serum and eluted it from different recombinant VAR2-CSAs in sequence and then changed the order around. I just want to quote something from from the abstract. It says, in 2 independent antibody purification depletion experiments with permutated order of VAR2-CSA variants, IgG purified on the first VAR2-CSA antigen displayed broad cross-reactivity to both recombinant and native VAR2-CSA variants, and it was functional. It inhibited binding of all isolates to CSA. So that's telling us, very similar to the data that Kevin Marsh and Russell Howard produced looking at the surface of agglutinating antibodies to the surface of infected red cells, with the VAR2-CSA with immune serum, we're seeing the same thing. The immune serum contains antibodies to a conserved epitope. Thus we can say immune serum of children contains protective antibodies against recently exposed strains, which are variant-specific, but not cross-reactive antibodies. Variant-specific antibodies are short-lived, and cross-reactive antibodies take many years to develop, but are long-lived with constant exposure. So this is the sort of cartoon I've drawn that sort of basically summarises that. You're exposed to various strains, you develop short-lived antibodies to that particular strain, and ultimately you'll develop antibodies to a conserved, by the time you're an adult, you'll have developed antibodies to a conserved epitope. And then if you leave that area and infection, you're no longer exposed to the parasite, those antibodies will eventually wane, and then you'll lose your immunity, but it can come back due to memory B cells to that conserved epitope. So what about vaccines with VAR2CSA now? So there's 2 vaccine studies published, as you know, and they both give similar findings. The PrimVac study from Serum et al. Demonstrated that when you vaccinate with the NF4 strain of VAR2CSA, the PrimVac antigen, you develop antibodies to the NF54 strain, but you don't develop antibodies to these other strains, 78 or FCR3. So what we're seeing with the recombinant is very different to what we're seeing with natural exposure. And the reason I think for that is fairly straightforward. This is the first time people have been exposed to this variant. I would predict that if they were exposed to multiple recombinant variants over a series of several years, they would all develop antibodies to a conserved epitope. Now, this is not just happening with the surface of the red cell antigen, it's happening with merozoite proteins as well. Now, maybe RH5 and AMA1 will be different, but the AMA1 data, which has been presented already by Prakash, suggests not so far, and MSP2 same. Strain-specific non-cross-reactive immunity has been shown following vaccination with merozoite proteins, strain-specific non-cross-reactive immunity. So it seems that so far we know with the merozoite proteins and the surface of the red cell is the same story. Thus, continuing on this analogy or this summary, unlike natural immunity, a whole protein vaccine will induce antibodies to immunodominant epitopes by definition. These antibodies will be strain-specific. So, subunit vaccines therefore need to be either a polyvalent vaccine containing immunodominant epitopes representing most strains or all strains in the community, or the vaccine needs to contain a protective cross-reactive epitope. The question is, how do you define cross-reactive epitopes? That's the question. So let me tell you how we've done that in a different organism, Streptococcus, group A Streptococcus. Streptococcus, as many of you would be aware, has a major virulence protein, the M protein. And when you're exposed to one strain of strep, of which there are hundreds and hundreds of different strains of strep, when you're exposed to one strain of strep, you develop antibodies to this immunoterminal epitope up here, and that gives you protection against that strain, but not against other strains. It's like malaria in kids. It's the same sort of thing. We decided many years ago now to look in this highly conserved region down here near the cell wall, almost buried, but not quite buried, and we looked at the C3 repeat region, the most conserved region of this entire protein, and we asked, did people who had strep exposure, adults, we've all had strep essentially, but if you're an adult, did you have antibodies to any peptides in that region? We made a series of peptides overlapping that region, and we looked at initially Caucasian adults from Brisbane in Australia, living in a strep non-endemic community or area, but all would have had a streptococcal exposure at some stage in their life, as children, if not as adults as well. None of them really made antibodies to any of these peptides. So you say, well, there's nothing there to follow maybe as a vaccine candidate, but I assure you there is. How do you find it? So when you now go into a streptococcal highly endemic community, and that's been defined as adult Aboriginal Australians who live in one of the most strep endemic regions of the world, have the highest rates of rheumatic heart disease reported globally due to post-streptococcal sequelae, you get a very different picture. What you see now is the very first peptide, peptide 145, is recognised by 95% of adult Aborigines living in this part of Australia where strep is highly endemic. These antibodies develop slowly over time. So if you look at young kids, very little antibody response. As the kids get older, they're starting to develop antibodies. So it takes years and years and years of exposure to strep to bring out this antibody response to this highly conserved epitope. Now, the question is, well, can we make a vaccine out of that? And I'm sure you know the answer because I wouldn't be showing you this data otherwise. But when we've tested this in mice, and currently it is in a human trial in Canada, but when we tested it in mice, we were able to show that if you vaccinate mice with that peptide, you can get protection, 95 to 99% reduction in group A strep bacterial burden post-challenge, whether the mice are challenged by the upper respiratory tract, intranasally, or by the skin. Looking at streptococcal burden in the spleen or blood, you get very, very good protection. So that epitope is conserved. it may be a bit like the RH5 that Simon was talking about. Because you're not exposed to it naturally, it is conserved, but once you identify it, it works. And we'll find out whether it works in humans, of course, when the trial is completed. Now let's just get back to malaria. I just want to share one data slide from Stephanie Yano's group in Edmonton in Canada. Stephanie has got working on an antibody 3D10, and she's found that looking at the ability of that antibody raised against PBDBP to inhibit binding of heterologous strains of falciparum to CSA. And what she's found was multigravid serum, as is well published by a number of groups, multigravid serum can inhibit binding compared to primigravid serum, but this monoclonal antibody, which is to a cryptic epitope clearly, can inhibit binding to a number of strains. So what Stephanie is trying to do and her lab is trying to do now is to find that epitope which is recognised by that monoclonal antibody. Now, I want to switch gears now, but talk about targeting conserved antigens, not of antibody-mediated immunity, but of cellular immunity. So, I've been working on cell-mediated immunity to malaria for a long while, and really inspired initially by some of the work from Bill Whiteance and his group, and Carol Long, of course, working with Bill. And we got interested in cellular immunity when I came back to Australia from working at the NIH with Lou. And what we found somewhat early on in the piece is that if you give a mouse a subpatent infection, they will make it very strong and keep it subpatent. So you infect them for 2 days, drug treat them, no parasites detectable by microscopy. Their spleen cells will respond very strongly to parasite in vitro. Differently, however, if you give a mouse a patent infection, their T cells from those spleens do not respond at all. And in other papers and elsewhere, we've shown it's due to apoptotic deletion of those T cells, and it's mediated in part by interferon gamma, and that's all published. So that was something that said to us, well, let's try that in humans, and we've worked with David Pombo, who was the first author on this paper, and David was able to demonstrate that following a subpatent infection of a number of volunteers with P. falciparum, all of them made a good Th1 T cell response measured by proliferation or interferon gamma response, and also induced nitric oxide synthase in all of them. So these people these volunteers who never had malaria previously made a very strong response to malaria as a result of a subpatent infection. So that led us to ask, well, could we induce that? And this is interesting to us because the theory was, or the hypothesis was, that a patent infection led to apoptosis of T cells, and that's to the parasite's advantage because T cells can kill a parasite. So the parasite wants those T cells to be ablated, and it does so as a result of cytokine-mediated apoptosis, as I mentioned. But if we keep the parasitemia low, well, we might be able to induce a protective, long-lasting cellular immune response. So the first way we tried it was in mice, and we, using a drug called centanamycin A, which was developed by Moses Lee, initially as a treatment for cancer, but has been used by Stephanie and other people in malaria parasites as well. We asked, could that drug attenuate the blood stage parasite, and we used Shibodi to start with, and if we take a low number, and when I say a low, we've gone as low as 10 to the 4 parasites, a low number of parasites attenuated in vitro with that drug, the drug's then washed out, you then give those parasites to mice, and you ask them later on, you give them 3 injections, although we have done it with 1 as well, are they protected? And they're protected against both the homologous parasite, a Shibodi vaccine, to Shibodi challenge, and heterologous parasite, Shibodi vaccine, Uelliide challenge. So they're strongly protected. So of course we wanted to do this in people. And so we're just finishing off analysing the data from a pilot study where 5 volunteers were vaccinated with about 3 million magnet-purified schizonts attenuated with the related drug to furamycin A. It's like centanamycin, a little bit different. 78 strain parasites, 4 weeks between injections, and challenged with 1,800 78 P. falciparum parasitized red cells 4 weeks later. And Danielle Stanisic in my lab was leading this study, and this work is now being just sort of getting prepared for publication. But what we found was that 2 of the volunteers out of 5, it was only a very small study, 2 of the volunteers following challenge did not develop malaria at all. Interestingly, they had symptoms of malaria, symptoms which suggested that they might be getting malaria. Some of them had some sort of a mild fever, I think one of them had, and we're still investigating that, but none of them had parasites that we could detect by PCR in the blood. Looking at the immune response, none of them had antibodies, and this is what we saw in the mice too, in Djibouti. They don't make antibodies. These people, the ones in colour, the red and blue, were the ones who were protected. They were protected in the absence of antibodies. And looking at for the cellular response, very small numbers, so we can't really get any correlates of protection, but we did notice that NK cell, TNF positive cells increased during the course of vaccination in those 2 volunteers who were protected. There's a slight hint that the interferon positive CD3 T cells may have increased as well, but this is the most interesting one here, the TNF positive NK cells. Not sure what exactly that means, but we're obviously following that up. But can we take a vaccine like this to a large-scale trial and develop a vaccine for clinical use? Sadly not, because there are major challenges with that vaccine, and one of them is how would you cryopreserve the vaccine? Even though you need very few red cells in the vaccine, they've got to be intact. The red cell membrane has to be intact. It's very hard to cryopreserve trophs. And how are we going to stop the induction of antibodies to human red cell antigens? And that's obviously a major issue. Even if you use O-negative blood, you can get antibodies to little c or little e. And we've actually seen that in various volunteers, not in that study I was showing, but in other studies. So we've gone to a liposome approach where we replace the red cell membrane by a liposome, and the kill parasite is in place inside the liposome. We use various ligands on the liposome, a mannose peptide F3 to target T antigen presenting cells, and we include 3D Fad, a synthetic version of monophospholipid A, to drive a TLR4 mediated Th1 response. So that's what the vaccine looks like, and what we do see protection in the animal models. So we've looked at Uelii here. So these are the vaccines where, because it's made in liposomes, we can lyophilise it down to a powder. And when we do that, we just reconstitute it of water prior to injection. Whether it's fresh or lyophilized, that vaccine gives you good protection. The mice all survive. But what about the problem of red cell inducing antibodies to the human red cell antigens? So we're tackling that by modifying the surface of the infected red cell prior to freezing it and putting it in liposomes by using methoxy polyethylene glycol, MPEG. So MPEG binds to, activated MPEG binds to lysines on the surface of the red cell, all antigens, not just red cell antigens. Any malaria proteins there, it'll bind to those as well. And then the PEG just basically camouflages the whole lot and stops the induction. In this particular study, which was published in 1997, it stopped it made that blood impervious to attack by antibodies to red cells. They were looking at developing universal donor blood. We use it, as I said, to try and make a vaccine. So how do we make this camouflage falciparum vaccine? First of all, it's a very simple recipe. You grow the falciparum in O-negative blood, so you start off O-negative to reduce the chances of getting red cell antibodies. You magnet purify them to near 100% pure trophs. You treat them with activated MPEG. You wash the free MPEG. You freeze and thaw it several times to kill the parasite completely. You bring it up to the correct concentration. You formulate it with liposomes and FAD, and you lyophilise or freeze. That's your vaccine. So that's what we're doing now in the human study, which is I'll tell you a little bit about in a moment. But when we asked, were mice, would that vaccine induce, stop the induction of antibodies to human red cells in a mouse, which is a zeta genetic difference, you see the unpegylated vaccine, whether it be fresh or frozen, they made antibodies to the human red cell. The pegylated vaccine did not. So that process of pegylation was able to stop the induction of antibodies to those red cell antigens. What about protection? Did it affect protection? We went back to the Uelii model and showed that the pegylated vaccine here in blue was as effective as the unpegylated vaccine. So the fact that the malaria antigens on the surface of the red cell were also pegylated, did not affect the activity of that, the efficacy of that vaccine. So right now, the M-pegylated camouflaged P-falciparum liposomal vaccine is undergoing formal tox for a human trial. So in summary, malaria survives in nature because its member parasites encourage the immune system of children to respond to immunodominant epitopes, which are not essential to the parasite species survival. The human immune system has not yet evolved to be able to quickly recognise cryptic epitopes. It takes many, many years of exposure for those epitopes to be recognised. The parasite is sensitive to cellular immune responses, but has evolved to use the host cytokine responses to delete parasite-specific T cells when parasite density reaches a critical threshold. But novel vaccine strategies using cryptic epitopes and low doses of whole parasite approaches to induce immune responses that the parasite has not yet evolved to counter may lead to a successful vaccine. And with that, it remains for me, of course, to thank the many people who have contributed to that work over the years. I mentioned Danielle and people working in my lab under her supervision. Other people, I've mentioned work from Stephanie's group in Alberta. Doctors at the hospital have done the clinical trials for us as study participants. Steve Hoffman and Kim Lee at Scenaria. All the other people mentioned on this slide. And of course, the funders who are mentioned there as well. Thank you very much.
Thank you. Questions? Can't see. It's really the end of the day. Third question here. Michael.
Michael, thanks. Great talk.
Simon.
Lots to think about. I was thinking about these cryptic epitopes that may be on PFEMP1. You inferred they may be on VAR2CSA. Is there anything from in silico or structural analysis that might indicate where
they are?
Where they are. I mean, in viral glycoproteins, they're sort of membrane proximal. You hinted that possibly with your group A strep.
Well, I'd say, Simon, you'd obviously want them to be surface exposed and you'd want them to be highly conserved. I mean, that would be where you'd start, I think. And then you'd have to, I don't know if there's any way to identify them other than empirically making peptides, whether they be conformational peptides or linear peptides, and seeing if they're recognised by, or seeing if they can induce antibodies which will recognise the parasite.
Well, just a big map campaign. I just don't think anyone's ever done it in the malaria field. But it's no different to what they've done on HIV and screening for BNAbs.
Well, that's what the Anna laboratory is doing with that antibody, 3D10.
Okay. Other questions?
Hello, Ben Sumner, NIH.
Okay.
Oh, up here.
Where are you?
Sorry. Gotta get under a light.
Okay. Good. See you.
So this might be a little crazy. What if the Scenario vaccine, not the just irradiated one, but the one that kind of goes through a few replications in the liver and then drops dead, would it be possible and or safe to make it kind of get to the blood stages and then drop dead, but not be able to replicate once it's in the blood?
So, yeah, thank you. So we've tried that actually with using apical knockout drugs like azithromycin. So you treat cells with azithromycin and, of course, or doxycycline, azithromycin. They'll go through 2 cycles and then they'll drop dead. So we have tried that, and you can certainly get induction of parasite-specific T cell responses, but I think it is an approach that we've considered. There may be other ways of doing it, but knocking out apical is one way that comes to mind. I think Joe DeLisi and others have sort of mentioned that as well, championed that idea.
Cool, thank you.
Thank you. Any final questions?
Thank
you, Michael. Thank you to all the speakers this afternoon and to all the speakers all day for a great symposium. Prakash?
Yeah, I won't take too much more time. I just wanted to thank a few people. First of all the speakers who've traveled near and far to come to this symposium and making this such a huge event and a success. Thank you all for doing that. All the participants, and I hear, we have had close to 300 in-person attendees and over 900 Zoom participants. So that's all thanks to the organization of Trish and Anna, who've been very instrumental. I don't know if they are here probably not here, but still a round of applause to them. I also want to thank Peter, right? I mean, he's been, a strong supporter for malaria research, and this is a symposium that's been going on for the last 13 years in this format, every World Malaria Day. I hope we will continue this, as Peter, is stepping away from the directorship, and we have a new incoming director, Dr. Jane Carlton. Unfortunately, she couldn't be here due to a prior engagement. But we are hoping to continue this meeting in the future years as well. But thank you, Peter, for all your support for malaria research, for young investigators, and your leadership and your enthusiasm has been much appreciated. Thank you for all your efforts.